Hello friends, I am Shirvano from TutorCabin Private Limited. Today in this lecture we study about the experimental evidence which is based on the semi-conservative way of DNA replication. This is our part 3.3. Our last two parts we discussed on the replication process and as well as the way or models of the DNA replication. We discussed three models of DNA replication. First is conservative, dispersive and semi-conservative. So this is an experimental proof given by the Matthew Michelson and Franklin Stahl in 1958. They performed this experiment, and by after the completing on the evidence, they proved that DNA replicate by semi-conservative way. The steps of this experiment is, first of all, they grow E. coli in the medium containing nitrogen isotope 15 as a nitrogen source for many generation. Clear? First of all, we take E. coli. E. coli is a bacteria. Okay? It's a gram-negative bacteria. Clear? Pink color. Observe in microscope. Rod shape. Rod shape. It means it is a bacillus. Clear? This bacteria replication time is 20 minutes. Clear? So, first of all, they grow in the medium containing nitrogen isotope 15. Clear? Clear? Because all the bacteria, all the organism needs to grow well some particular or appropriate medium. Clear? Nutrition medium. This is a nutrition medium. It is the best nutrition medium for the growth of bacteria. Clear? But they also contain the nitrogen isotope 15. Isotope 15 is a heavier nitrogen isotope. Clear? Because DNA contains a nitrogenous bases. That's why this nitrogen is absorbed by the bacteria. Clear? So the bacteria DNA contain also and 15 nitrogen in it. These E. coli grow well. Clear? After the absorption, clear? DNA are extracted from the bacteria and then centrifuge it. Clear? The help of apply of cesium chloride. Cesium chloride apply for as a density gradient. This heavier DNA molecule, because N15 is heavier isotope. Clear? And N14 is lighter than it. Clear? So N15 is heavier. So if the DNA absorb the N15, they become a heavier DNA. Clear? That's why scientists said here, this heavy DNA molecule could be distinguished from the normal DNA. Clear? Normal DNA have not contain the N15. Okay, so both are normal are lighter than N15. Sorry, so N15 is a heavy isotope because they bacteria absorbed it, they become a heavier. Clear? So this heavier DNA could be distinguished from the normal DNA by centrifugation method in a cesium chloride density gradient. If we make in our home chutney or some sauces, we make some ingredient plus water. The water is in place of cesium chloride here. Clear, friends? So the cesium chloride mix it, and if we centrifugate, we saw in the test tube that contain the supernated plus some some DNA palette. Clear? The supernated contain only cesium chloride, but the palette contain DNA contains nitrogen N15. Clear? Scientists understand by this process, they learned the DNA absorb the nitrogen. Clear? They become heavier than before. Clear? So in this way, scientists Michelson and Franklin Stahl N15 nitrogen E. coli, these E. coli transfer into the medium containing, clear, medium containing N14. Clear? Both are isotope, but this is a 14 point. Clear? This is a 15 point. So in both of them, this is a heavier. Okay, so this is a lighter and this is a heavier. Heavy isotope and light isotope. Clear? It's a lighter and this is a heavier. Clear? So 20 minutes is the time for replication of E. coli. E. coli are replicated and divided in 20 minutes. Clear? E. coli transfer. Clear? Clear? E. coli contains, because previously E. coli contained N14, but in this way E. coli contain also 14. Clear? After 40 minutes, this process are continue, get the result by this way. Clear? Clear? This tube contain only supernated plus DNA. It means the band is present only here. Clear? And 15 band. After 20 minutes, tube contain 2 bands, one hybrid band. Clear? One hybrid band. This way. Clear? After 40 minutes, two bands. And 14 and hybrid. This way hybrid. Clear? Clear, friends? This is a first step, this is a second step. Clear? After showing the electronic microscope, scientists saw that this way. Clear? These contain 15 nitrogen. After 20 minutes, clear? After 20 minutes, why we wrote 40? Because 20 plus 20 total 40 minutes. In our NCERT book also write this 40 minutes. So 20 plus 20 calculated the 40 minutes directly. Clear? So these type of two hybrid bands of DNA. Clear? These hybrid bands. Clear? After 40 minutes, they calculate that, observe that some two equal number of lighter as well as hybrid bands are present and observed in the electronic in the microscope. Clear? This is a N14. This is a hybrid lighter N14 lighter. Clear? N14 15. Clear? 15 14 hybrid. Clear? The N15 only heavier. Clear? This observation 40 minutes. Clear? This diagram is also given in the your NCERT textbook, figure number 6.7. So the second step, then they transfer the cell into a medium which normal 14 and took sample at a various different time interval. The cell multiply and extracted. The DNA remain as double strand. Clear? They transfer it several intervals. Clear? Centrifugate, extracted the DNA by this method. DNA extracted and centrifugation by the adding density gradient sodium chloride. Clear? So these bands. Clear? The DNA that was extracted from the culture one generation. This is a first generation. Clear? First generation, and this is a second generation. Clear? This is also a first generation, this second generation. Clear, friends? N14 to N15 medium, that is after 20 minutes, E. coli divides in 20 minutes. Clear it? Hybrid or intermediate density. So we saw this hybrid and intermediate density because N14 is lighter than N15. Both densities are different. N15 are highly dense. That's why they settle in the bottom. Clear? The DNA extracted from the culture after another generation, and this 40 minutes, second generation, this 40 minutes, and was composed equal amounts of this hybrid DNA as well as lighter DNA. There are two bands are observed here. Sorry. This is a hybrid band. Clear? This is a hybrid band. Clear? So equal number of hybrid as well as lighter present in these medium, this generation, second generation. Though the scientists result it's observe the equal number of hybrid and lighter band were observed. Clear? So the conclusion is, the result is by this, it is hence proved that by this experiment, hybrid DNA, equal number of hybrid as well as lighter DNA are present. The DNA replicate semi-conservative way. It is proved by this experiment, Matthew Michelson and Franklin Stull, experimental evidence. Clear, friends? So, this step please write down and also draw this well-defined diagram. So, it is a clear E. coli is an organism, gram negative, observed in pink color after gram staining. So, it's a bacilli, rod shape. This replication time or division time is 20 minutes. First of all, they cultivate in the medium containing nitrogen is 15. Clear? The extracted after cultivation extracted DNA and centrifuge it the density gradient. The scientists saw that the DNA, normal DNA are not contain heavy, it's a lighter than these DNA. So this DNA contain N15 as a isotope. Clear? It's become a heavy, lighter to heavier. It turns lighter to heavier. After there is a step, sorry, this is the second step. This is a third step. Clear? The first step is after this N15 E. coli transfer into N14, they absorbed also N14 and these follow the several generation, and after these centrifugation are take place here, and also examine these bands in the with the help of techniques of visualization techniques. So the scientists observe these bands, equal number of lighter as well as hybrid bands are found here. So the result is equal number of hybrid band and lighter bands are observed, and the hence DNA replication follow the semi-conservative way. Clear? Hope you understand in this lecture. Hope this is easier to understand the experimental concept. But if you have any query, please ask me. Thank you.